Okay, good morning, everyone. I just hope you're all having a great week. This is the second lecture for Chapter 3, and today we'll be kind of continuing our conversation talking about the cellular levels of organization. Again, if we go back to think about that figure that we went over maybe the first or second lecture about those different levels of organization, we're slowly but surely working our way up. We started with the atomic level, next we went to talking about macromolecules, how those macromolecules can make different essentially organelles or be comprised of different organelles. And that's what we'll kind of continue talking about today, are the different organelles that make up the cells. And then we'll finish talking about cells, how cells divide, and some of the effects of aging on cells as well. So hopefully you all had a good weekend. I'm recording this on Monday morning. And so, yeah, just kind of a quick reminder, if you're watching this, please make sure that you are answering the study guide questions for Chapter 3. Your quiz 3 for Chapter 3 is due by midnight this Friday, so hopefully you have watched this lecture by this coming Friday. Like I said, I am recording this on Monday, and it's not on the list, but if you haven't done your adaptive practice for Chapter 3, that is due tonight on Monday. And then next week, kind of the following week on Monday, the adaptive practice for Chapter 4 is due next Monday. So just make sure that you're staying on top of these assignments. I know that there's a lot of things going on. I know that a lot of your professors maybe are using different websites and you're having a lot of different due dates. I really tried to make sure to remind you of these due dates pretty repeatedly, pretty frequently. It's listed on the Moodle site. We talk about it here in lecture. And if you're having a hard time kind of remembering them on your own, maybe putting them into your calendar or setting an alarm in your phone will also kind of be a really nice reminder as to when each one of these assignments are due. And again, everybody has to-do lists, and if that's one way to kind of help you keep on track for your to-do list, I would highly recommend doing that. Oh, something I want to quickly remind everyone, I've had people kind of reach out with issues a couple times over the last week or so about not being able to see things in Wiley Plus. Just make sure that you're using Google Chrome or Firefox, not Safari. Do not use Safari for Wiley Plus. And so if you're having any issues, that might possibly be why it is. Okay, so study guide questions. We'll be going over these on Thursday. Make sure you are being able to describe the structure and function of things like cytoplasm, cytosol, and organelles, and so on and so forth. So we'll go over these more in depth on Thursday as a group, and then we'll probably discuss them as a larger group either at the end of Thursday or at the beginning of Tuesday's lecture. Okay, so the structure of the cell. Sorry, my light goes off, so it kind of washes me out. For coloring, I mean, if I'm going to post this many videos on YouTube, I probably should just invest in a ring light like a real YouTuber. Okay, so structures of the cells. So far, the entire last lecture, shockingly, we spent a majority of our time talking about this thing that encompasses the entire cell, the plasma membrane. The plasma membrane is really important for transport, as you hopefully saw in that lecture. And again, passing things into and out of the cell, not only is kind of an import-export process, but it's also important for the cell's function. So maintaining things like ionic concentrations inside and outside the cell, maintaining osmotic pressure, the number of solutes inside and outside the cell kind of help maintain cell shape. So the plasma membrane really is kind of more than just like an in-out door mechanism. It really does help maintain cell function and cell viability, help that cells kind of survive and thrive. So today we're going to kind of play cleanup, and we're going to go through a bunch of the other organelles that are contained within the cell. So if you have not done so far, you can open up your own PowerPoint, click on the cell anatomy link, and that should send you to a nice video through Wiley Plus, and kind of going over, again, some of these different cell structures. And I'm not going to spend a ton of time on this slide because we'll probably see it at the end of this presentation again, and we can kind of review everything that we have gone over. Okay, so just kind of reminding you of what we have talked about and kind of what we're going to go over today. So although it's not here, shockingly, I don't know why, the cytoskeleton, I think is the part, skeleton, should be written here. The cytoskeleton is basically kind of like, you can think of like the tent poles of the cell. So if the cell were a tent, it's got that, very fluid plasma membrane, kind of like a tent is very fluid. The cytoskeleton is composed of a bunch of kind of tent poles, and those tent poles kind of have different, let's say, sizes. So the three type of tentpoles you have here are things like microfilaments, intermediate filaments, and microtubules, and they kind of allow the cell to have different structures. So we'll see kind of at the very end here the different cellular structures that are seen within the human body. It's pretty amazing that all of these cells are able to kind of have that same blueprint, but have all these different cell structures, and the cytoskeleton is what assists them in having those different cell structures. So cytoskeleton is very important for the structure and the function of those different cells. Other organelles that we'll talk about today, the centrosome is important, the pair of centrioles plus the pericentroidal matrix. This is very important for mitosis, which is cellular division. So when we get to talking about how cells essentially replicate and divide, and if you have a single cell and then it becomes two cells, kind of the centrosome is very important for that process. It also kind of attaches to some of those parts of the cytoskeleton, things like tubules. And so they're kind of like anchors on each side of the cell that help the cell maintain its structure, but then also aid it when it's going to divide and part ways and kind of take one cell and become two. Cilia and flagella, if they've watched the last lecture, I think they're important for motility. So if a cell needs to either move as an individual cell, or if it needs to aid the movement of, let's say, liquid around it, the cilia are very important for that. And so a lot of the cells that line our mucous membranes or our intestine are going to have the cilia because they're going to try to be moving things along. So you might have cilia in an area that has, mucus, things like your nasal cavity, or if you think about your intestines, it's important for things to be kind of kept moving through those areas. And so cilia are kind of expressed on the surface of those cells or hanging out on the surface of those cells. You just kind of keep moving things along over that cell surface. Flagella, like I said, we don't see that nearly as frequently in the human body. We don't see cells with flagella on them. But they're going to move the entire cell. And you see those in single-celled eukaryotic organisms, again, which we are not. Okay, other cellular organelles, ribosomes. So I introduced the idea of ribosomes in the last lecture. Ribosomes are basically little protein factories. So if we talk about how DNA gets made into a protein, ribosome is really kind of where that process takes shape, where DNA, I guess I should be using the draw, DNA becomes RNA, and we haven't talked about this process and we probably won't, becomes protein. This is what's called the central dogma. It's very critical for cell biology. So if you're taking any kind of cell biology class, you'll probably go over this. But kind of how does this process actually happen? What organelle actually makes this happen? And the answer to that is a ribosome. A ribosome basically interacts here between the RNA protein step, and it's the organelle that actually makes that happen. So you've got two subunits. A large one and a small one. And basically mRNA kind of goes through there, gets read, and then a protein gets made out the top. That's a very crude picture of it. And since we're not going to go into it any more in depth, I don't necessarily expect you to recreate it, but it's just a very kind of raw caricature of what happens during protein synthesis. And if you're more interested in that, I'd be happy to talk about it. Ribosomes, if you remember, stud the rough ER, and then also can be freely floating through the cytosol. So you can have proteins being made in different locations of the cell, not just on the surface of the rough ER or endoplasmic reticulum. Okay, which follows the next thing here, the endoplasmic reticulum. So it's a membranous network of flattened sacs or tubules. So if you remember kind of that image of that first figure showing the large cell. And then since it's rough ER, there's going to be all these ribosomes studding the surface of it, and that's going to be essentially making all of that protein. It's covered by, the rough ER is covered by ribosomes. The smooth ER lacks ribosomes. They can synthesize glycoproteins. So glyco meaning, sugars. Proteins, so proteins that have sugars attached to them, and phospholipids, and that's basically a fat with a phosphate group attached to it. If you remember, someplace we might find phospholipids, that would be the plasma membrane. So it's important that we make more phospholipids to insert into the plasma membrane. They can also be secreted during exocytosis. So last time we talked about how exocytosis is when something is released from the cell, and endocytosis is when something is brought into the cell, and that's through the process of vesicles. So the vesicles are just kind of these smaller little pockets of membrane that can either be released to the outside of the cell or sometimes bud and come into the cell. So endocytosis in, exocytosis out. And we'll talk about that when we talk about, you know, types of cells that secrete things within the human body. Synthesize fatty acids and steroids. Some of those have signaling mechanisms in the human body. Inactivates or detoxifies drugs. Removes phosphate groups from glucose 6-phosphate, important for metabolism, and stores and releases calcium ions from muscle cells. A very important function for muscle cells to be able to release those calcium ions, because that's essentially what's signaling that muscle cell to contract. So we'll talk about that a lot when we get to the chapter on muscle cells. So understanding that when we are talking about calcium being released from the endoplasmic reticulum, this is the organelle in which that's derived from. So just, I know most of you are sitting here saying, oh, why are we learning about the cell? But the cell is an amazing thing. And again, the cell has a very direct correlation to a lot of the different tissue types that we'll be talking about. So it's a great example there, how the ER stores intracellular calcium is an important signaling mechanism for muscle contraction. Okay, the Golgi complex consists of 3 to 25 membranous sacs called cisternae. They look kind of like the ER, a little bit structurally and functionally divided into the entry and exit. Golgi complex, you can think of almost like the postal service of the cell. It's going to be kind of sending things out and bringing things in. So when we talk about the process of exocytosis and endocytosis, that's very much mediated by the Golgi complex. So it's accepting proteins from the RefER, and then from there, it's going to modify the molecules further, and it's going to sort and package them into their transport for their destination. So again, Golgi complex, very much think mail service. I mean, it's, again, like the cell is basically a protein making factory. And so, you know, you can think of the ER as being kind of the production line, and then the Golgi complex being the shipping department. Okay, lysosome. Lysosome would basically be, if you were talking about the cell being a protein making factory, it would probably be your custodial staff playing cleanup. So a lysosome is a vesicle formed from the Golgi complex and contains digestive enzymes. And so if you say, let's have a type of protein that maybe is misfolded or isn't carrying out the function that it should, it's going to be shuttled to like a lysosome, which is another type of vesicle, and we can think of vesicles as kind of substructures, very similar to like a whole cell, but within just a tiny part of the cell, and it's very acidic in there. What the lysosome does is, because it's very acidic, it's going to break down that protein into its original components, and then it gets kind of reused by the cell. So it digests the contents of endosomes. Again, that's bringing those vesicles into the cell. Phagosomes, where the cell kind of goes and eats a component of the outside. White blood cells would be an example of something that would phagocytize an outside component. And vesicles form during bulk phase endocytosis, which we don't really need to go into the specifics of that, but again, another type of endocytosis. And transports final products of digestion into cytosol, digest worn out organelles, entire cells, autolysis, and extracellular materials. But a lysosome basically just plays cleanup for the cell and degrades everything using its highly acidic vesicular environment. And then each one of those components can be reused by the cell later. So you can definitely think of the lysosome as playing cleanup for the cell. Okay, a peroxisome, it's very similar kind of to a lysosome, except for it's using oxidases, are oxidative enzymes, and catalase, which is a hydrogen peroxide. So instead of just using acid, you've got these other types of oxidation reactions occurring here. So it's going to oxidize amino acids and fatty acids, detoxifies harmful substances such as hydrogen peroxide and associated free radicals. We talked about free radicals when we were talking about kind of atomic structure. So this basically kind of plays cleanup for things like the free radicals and hydrogen peroxide. So it's a little different than a lysosome, given kind of the mechanism at which it carries out its function. The proteasome is going to also kind of play cleanup for the cell. Here it's a barrel-shaped structure, so it literally looks like a barrel on the molecular scale, and you have proteins that go into it, and the proteins just basically get shredded up back into amino acids. So if you remember that proteins are composed of amino acids, the proteasome just kind of degrades them all back into amino acids. So it's going to degrade extra proteins that maybe aren't needed. It's going to degrade proteins that are damaged in some way or maybe not made correctly. And it's going to cut them into tiny peptides or back into amino acids. And so those amino acids can go back into the cell and then maybe get used to make another protein. So a proteasome basically is like a little paper shredder, except for it's not paper that's being shredded. It's proteins that are being shredded. Mitochondrion. So mitochondrion, hopefully most of you have, heard of mitochondria, ATP production, which is the major energy source for the cell. And so the mitochondria, everybody calls the powerhouse of the cell, consists of external and internal mitochondrion. Cisternae and matrix. Mitochondria are interesting because they have their own DNA as well. So the mitochondria kind of looks like this, and it's going to have both an internal and external membrane. Those differences in those membranes is going to be important for carrying out the function of creating ATP. I don't think we'll go into the specifics of that, but I could be wrong. But interesting thing about mitochondria is, like I said, they have their own DNA. They're kind of their own little entity. There's a hypothesis that mitochondria were once their own single-celled structure, and a eukaryotic cell said, hey, that single-celled entity makes a ton of energy. Why don't I engulf it, and we can kind of help each other out, and I'll protect this smaller substructure, and it will produce a lot of energy for me to survive and thrive. And so that's called the endosymbiotic theory, meaning they both got something out of it. And so again, mitochondria makes a ton of ATP through aerobic cellular respiration, meaning that it requires oxygen, and plays a role in apoptosis, which is when the cell basically says, hey, I'm done here. I'm going to basically self-detonate. Maybe we'll talk about apoptosis at some point in time. So mitochondria, powerhouse of the cell. The nucleus, again, kind of thinking of the cell as a protein-making factory. This is probably kind of like maybe the R&D department, or where all the blueprints or all the ideas kind of come from for the cell. So if we look at the nucleus here, it contains DNA. DNA is the genetic material that's passed from cell to cell. It has a genetic code associated with it. That genetic code codes for all of the proteins that are made in the cell. It codes for kind of how those proteins are read and what their functions are, and then what that cell is specifically made to do. The nucleus also, outside of just housing DNA, has pores, nucleoli, chromosome, which is basically packaged DNA. So chromosomes is just packaged DNA, exists as a tangled mass of chromatin in interphase cells. So chromatin is just, again, a DNA that's been tightly packaged. We won't go into the specific of how that's tightly packaged. If you would like to talk about more, I'm happy to do so. But the nucleopores control the movement of substances between the nucleus and the cytoplasm. So DNA and RNA have to, or DNA not, but RNA has to exit the cell at certain points, again, to be read by ribosomes and to make proteins. The nucleoli produce ribosomes, and the chromosomes consist of genes that control cellular structure and cellular structures and direct cellular functions. So that's kind of, again, the major component or the major purpose of the nucleus is to direct structure and cellular functions. So just to make it very clear, DNA is just that double-stranded thing that we think of. We, as humans, we have 23 23 chromosomes, which is basically 23 pairs of DNA stretches. Those DNA stretches kind of get tightly wound, and that's what's called chromatin. So I know you're seeing words here, things like chromatin, things like chromosomes, but for both of those, for our purposes, just think that that's basically the packaging of DNA, and that's the purpose of the nucleus, is to kind of house that DNA, and it's its own membranous structure. That DNA is kind of kept separate. Okay, so the cytoskeleton, like we introduced kind of that first in those first few slides, we talked about how the cytoskeleton can be the tent poles that help maintain the structure of different cells and different cell types. And a great example of that would be this endothelial cell that you see here. You'll notice that this cell is not the regular kind of round cell that you think of that we saw in some of the previous figures. This one has a very specific structure. You've got these microvilli that are on one side. You'll notice that this cell doesn't look the same on both sides. It has two different sides, and we'll go over what those sides are called, basal and apical. But the cell still has a nucleus. But those microfilaments you can see here are very small kind of tent poles. So they're giving that cell very specific structure and kind of the smaller protruding microvilli that are extending out there. So microfilaments are kind of the tiniest type of tent pole that you can use for the cell. Kind of the larger, medium, I guess I should say medium, the medium-sized tent pole that you can use are called intermediate filaments, and those are, you can see here, maintaining kind of this middle structure of the cell. They're not extending out into these microvilli. You can see pictures of them here. These are wonderful, EM pictures. Scientists get really excited about these when they take these pictures, because they're like, ooh, these are the things that you learn about in textbooks, but this is what it really looks like if you took a picture of it in a cell. And so intermediate filaments are kind of the medium-sized tent pole that gives you, not the super defined structures, but not the super kind of large overarching structures. And so the next size up would be your microtubule, which is your largest type of structure. Here you can see we talked about the centrosome, which is going to be important for cell division. And those microtubules are extending out from that centrosome and giving kind of the bulk, structure to that cell. So it's not super fine, it's not intermediate as intermediate filaments do, giving it kind of that bulky structure in the middle. This is kind of the largest type of tent pole that you would be using for your tent, kind of anchoring things down. This anchors to the centrosome, and so when the cell does decide to divide, this larger bulky anchor helps kind of the division of that cell into two different components. And so if you don't like the tent pole metaphor, which I like to use metaphors because it gives you a nice visual, because you've seen a tent, I hope you find that helpful. Okay, so we just learned about the centrosome here. That's located right above the nucleus, and the microtubules. If we kind of break down what those specifically look like, here you can see the centrioles themselves, and its arrangement of microtubules in the centrosome. This is just a picture of what those centrosomes look like, or the individual centrioles look like. Those are the kind of the individual, I guess, anchor pieces here. We're not going to probably talk about centrosomes or centrioles. The level that you need to understand is that they're important anchors for microtubules for cell division. It's probably the level that you need to know it. We don't need to go a ton into this, just because, again, not really the focus here. Okay, we'll see that cilia and flagella are, like we went over before, important for cellular movement. And so you can see that the cilia or flagella have here these microtubules that extend down into them. So they're important for maintaining that structure. because a flagellum is not a super fine structure. It's actually pretty large. One place we do see a flagellum, I guess I kind of discounted this earlier, one place you do see a flagellum in the human body is on a sperm cell. So the sperm cell is going to use the flagella kind of like in a rotary motion to project itself towards, let's say, an egg. And so it's got a tremendous amount of force that's generated through that kind of propeller motion of the flagellum. And so the way that sperm cells are able to do that is because they have these basal bodies that are attached to the main cell body of that cell, and the doublet of microtubules kind of working a circular fashion to propel that sperm cell forward. You'll also see that this cilia also have these types of cytoskeletal structures. And like I said before, it's important for cilia to exist on areas that have mucous membranes, or you've got fluid kind of moving through areas. And so this is a great image showing cilia lining the trachea. And so you can see those individual cilia that are going to be important for moving liquids, moving larger bodies, kind of, let's say, through your throat or through your trachea, and we'll talk about kind of cilia lining those other mucous membranes. Okay, so the movement of cilia and flagella, I guess I've kind of been hand gesturing these movements. Here you can see kind of the movement of a cilia. It's almost like a whip-like structure. It kind of, if you've seen probably a lot of cilia that, or things macro that look like cilia, probably in the ocean. If you think about certain types of, what am I thinking of? Things on the Great Barrier Reef. Oh gosh, algae. So you can see that it kind of works in a flicking motion, helping again, moving everything towards a certain specific direction. So you can see the recovery strip kind of goes back, and then as it goes forward, it's able to move, liquids or mucus in a very specific direction. Flagellar movement, kind of like I was showing you with my finger, it's very circular, almost like a corkscrew. So as it's moving, it's kind of got a corkscrew tail movement. That movement is powered by ATP energy, not shockingly, but there's a specific mechanism that kind of helps the cell maintain this flagellar movement. And again, the example that we talked about just a second ago was the sperm cell. So cellular movement is important for specific cells, or aiding the movement of things outside of the cell is also can be very important, as you can see here. Okay, so I introduced the idea of what ribosomes are earlier in that chart, and again, they're just little protein making factories. So they make proteins from RNA. So RNA makes proteins. And so that's what ribosomes do, is they basically step in here and allow this process to take place. So we can see it's zooming in onto the rough ER here that is studded with ribosomes. There is a large subunit, there is a small subunit, and those subunits come together to form a completely functional ribosome. And so the mRNA or the RNA comes in and is read, and then a protein basically comes out the side. And so you can see here the rough ER of ribosomes. Sorry, not the rough ER, this is the nuclear pore. So you've got pores on the nuclear membrane. You can see a nuclear pore here, but it's also studded with ribosomes. So the mRNA, as soon as it's being brought out of the nucleus, is being made into a protein. This is a very rough kind of schematic of what happens in this process. And again, if you want to talk about this more specifically, I would be happy to do so. The small subunit and small subunit and large subunit of ribosomes are different for eukaryotic organisms such as ourselves and prokaryotic organisms such as bacteria. And because of that, there are actually some drugs that you'll take for bacterial diseases that target the small and large subunit of that bacteria, because again, any place that the cells differ, whether from between a prokaryotic and eukaryotic organism can be a target for a drug, so you can selectively kill just the bacterium. So that's kind of an interesting, anecdote about the differences between ribosomes and how, as clinicians and physicians, we can utilize that is by targeting, the differences between them, making drugs that target just a small and large subunit of bacterium versus eukaryotic cells. It's a very great drug target. Okay, endoplasmic reticulum. So this is a great image showing the endoplasmic reticulum. They're little protein-making factories. We've talked about it before. They're located right outside of the nucleus. So you can see here as, say, DNA is becoming RNA. Wow, this drawing is not great. RNA, and this RNA is being shuttled outside the nucleus, and then as soon as it does, it's going through the ribosomes. You can see the ribosomes are going to be making the protein, and then from there, they'll be going over to maybe the smooth ER as well. So you see the rough ER here that has ribosomes on it, and the smooth ER that is lacking ribosomes. And so what those smooth ER could be doing is actually helping attach, let's say, sugars, so you can make glycoproteins. So if that protein needs some kind of like decoration or modification, as a scientist might say, the smooth ER is helping make that modification. So here we can see the smooth ER and the rough ER, kind of how those two differ. And again, scientists get super excited about these beautiful pictures because this is what it really looks like. It's not just a cartoon. You're seeing an actual image of what rough ER and smooth ER look like. And so trust me, there was a scientist one day that was sitting in front of a microscope, took this image and said, oh my gosh, that's so beautiful. It needs to be in a textbook, and here we are. Okay, so rough ER, smooth ER, next kind of in the whole protein factory kind of metaphor. Again, we've talked about the nucleus where the blueprints are located, how it goes from nucleus through the ribosomes for the protein to be made into through the ER modifications to the smooth ER. And then next we have the Golgi complex, which is like the kind of, we'll say, shipping center. Maybe that's a better way to put it. Shipping center for the cell. So here you have your shipping center, and your shipping center, kind of the way that the cell likes to package things, like how if you send something through the mail or through UPS or FedEx, you have to put it kind of in a package for them to acknowledge that it is in fact something that needs to be sent. If you put, like, let's say a book out on this on your mailbox, your mailman's going to look at you like, no, I don't do that. You have to put it in a box. And so very similarly, the Golgi complex has to package things into vesicles. And so here you can see kind of that vesicle. Here's another vesicle, and that vesicle allows that the contents of whatever's inside to be shipped to the outside of the cell, or maybe even be brought back in. So you can think of that vesicle kind of being as the shipping container that allows the cell to send things out or receive them. So the vesicle kind of goes from the rough ER, and this is called the cis phase. Cis and trans, man, it'll come back to haunt you multiple times throughout biology. And I don't know if we'll be talking about it in all the different ways it's used, but cis and trans, here you have your cis face or your same facing face. From the ER to the cis face of the Golgi complex. So this is kind of the direction things are going, and this will bud and kind of infuse and bud and fuse, and it's basically fusing and budding and fusing and budding as a vesicle until it hits this face, which they call the trans face or the exit face. So this is kind of like the last stop before it would go and bud with the membrane and be sent outside the cell. And so the cis face is located closer to the nucleus. The trans face or exit face is located farther away, more towards kind of the plasma membrane. So it's kind of just a directionality thing to describe the Golgi complex. So your exit face here, you could have a secretory vesicle exiting the cell that goes and merges with that plasma membrane and fuses with it, and then all of the secretory contents get released outside of the cell. So this happens with any type of, let's say, oil that is on your integumentary system. It gets released onto the surface of your skin to maintain, you know, that nice oily skin face. Or, you know, another place this might happen is in, let's say, your nervous system. Your brain cells also use this to release neurotransmitter and send these cellular signals. So the process of exocytosis is very important. We'll talk about it several other times. So here you can see kind of, again, another beautiful image of the secretory vesicle being released and fusing with that plasma membrane. Man, there are some beautiful images here. Okay, so hopefully you are keeping track of each step of the process and how this protein making factor is sending things out of the cell. This is another image. This is not a beautiful image that's taken from a real cell. This is obviously a cartoon where you're seeing, you know, your synthesized protein being made here in your rough ER via ribosomes, being put into this transport vesicle. It fuses with the cis side closer to the nucleus, and then it kind of buds and fuses through the Golgi complex. And then here, it has multiple different kind of paths it can take. It can become a transport vesicle, again, where it's budding and fusing with more of the Golgi complex. Here it's a secretory vesicle where, you know, the protein that was made is getting exocytosed or just put on into the extracellular environment outside of the cell. Here you have a example of where the final shipping placement of this is for the protein that's embedded into that vesicle to be embedded into the plasma membrane. So instead of being released out into the world, it's just going to fuse, and then that protein that's embedded into it is going to be hanging out in the plasma membrane. And then the last kind of example that we see here is a transport vesicle to a lysosome. That lysosome, again, is going to kind of degrade that vesicle, and that's a kind of a different process than what we're talking about here. Yeah, transport vesicles, some of those proteins can also go, let's say, not just away from the nucleus, but some of them go back into earlier structures. So let's say if you have a vesicle that needs to fuse, it kind of keep that larger face here, because if you think about it, it's budding and fusing and budding and fusing. If you kept doing that, you would lose the bulk of this size here, because you would keep continually just be pushing things out. So some of this, these vesicles actually have to recycle back or go back and bud and kind of help keep the bulk size of that first cisternae intact. So there's a lot going on in the Golgi complex, and that's basically like probably 4 lectures of cell biology right there, condensed down into a single slide. So don't feel super overwhelmed by this. Probably in this class, the thing that we'll talk about the most is going to be the secretory vesicle. We might talk some about these membrane vesicles that fuse with the surface of the cell membrane, but I don't think it's necessarily important that you know, memorize all of the steps of vesicular fusion, which is what this would be called for an anatomy and physiology class. But it's good to know that it exists and that there are these different kind of places that these vesicles can end up. Okay. Lysosomes, like I said, kind of like the trash can of the cell a little bit. They're going to be breaking things down. We'll talk about how lysosomes and peroxisomes are very kind of similar in that way. But lysosomes have these digestive enzymes, this highly acidic environment within it. So it's its own little pocket or vesicle within the cell. So, you know, you got this whole cell membrane here encompassing the cell, and we keep talking about vesicles, but those again are kind of little sub-blebs within the cell. And these digestive enzymes, these highly acidic pockets, are able to break down the internal contents of that cell. So if a protein has, you know, not carried out its function, isn't properly folded, sometimes it will get shuttled to a lysosome, it'll get broken down, and then it can be utilized again. Lysosomes and let's say proteases are slightly are different. Lysosomes are their own vesicle. Proteasomes are kind of their own protein structure, so they're not encompassed into its own double membrane structure like a lysosome is, like a vesicle is. Peroxisomes are structures that are similar in shape to lysosomes, but are smaller and contain enzymes that are used They use oxygen to oxidize or break down organic substances. This is just another kind of mechanism in which the cell breaks things down. The types of things that it targets to break down are different than what you see in a lysosome, but we'll just kind of keep it at that level of understanding right now. Peroxisome uses oxygen to break down some organic substances. Proteasomes, very, very similar. I keep saying how lysosomes are important to break down proteins, but they can also break down parts of organelles as well. They're much larger than proteasomes. Proteasomes are literally like a barrel. This is a terrible drawing of a barrel. They are very similarly, we'll go like this, a barrel structure. And this barrel structure, and there's multiple components to it, or subunits. But what happens with the proteasome is you've got a protein that enters as a full-on protein here, and then what comes out the other side is just basically little chunks of protein, or maybe individual amino acids, and then those amino acids can be recycled. Let's say they get recycled, then they would be the thing that we talked about just a second ago, was the ribosome. So there's a large subunit and the small subunit, and so it could get utilized by the ribosome to again make another protein. Make another protein. That's the way it would go. So proteasomes can be important for kind of the recycling of proteins. So they can destroy unneeded, damaged, or faulty proteins by cutting long proteins into smaller peptides, or what we might call amino acids. And so if a protein is kind of messed up, it can get sent to the proteasome, and the proteasome is going to degrade that protein and break it down. So it's kind of like the recycling center. We'll call it the recycling center of the cell. Okay, mitochondria. We introduced this idea of it being the powerhouse of the cell. The powerhouse of the cell has these double membrane structure here that you can see, and each one of these little pockets here are called the cristae of the cell. You can see that there's a mitochondrial matrix that exists in here. It's the difference between the internal and external parts of this cisternae that cristae, that allow the cell the mitochondria to utilize the differences in concentration to create ATP. It's part of its function, but we won't go into that a ton right now. You'll recognize that there are ribosomes located within the mitochondria. They'll make their own proteins. There are enzymes. The mitochondria really could be its own little entity. It's got its own DNA. It's got its own ribosomes, and it's making a ton of energy. But again, the purpose of mitochondria, the big takeaway from mitochondria is the fact that they make energy in the form of ATP, or adenosine triphosphate. Okay, here we are going over the nucleus. Again, this is where, the very important DNA is located. See here kind of these darker stretches, this annotated chromatin, and chromatin is just basically very tightly packaged DNA. So chromatin is, you have the stretch of DNA, your DNA gets packaged into chromatin, and those chromatin kind of, again, are situated by different chromosomes. But if you see chromatin or chromosomes, just think DNA. It's also a double membranous structure. You have a nuclear envelope, a nuclear pore, which allows, exportation of mRNA outside of the nuclear pore, and you also have the presence of polyribosomes, which are just multiple ribosomes that would allow for proteins to be made. And so you can see here how tightly correlated that rough endoplasmic reticulum is. So as DNA is made into RNA, that RNA gets exported out of the nucleus and can be made into protein. Sorry if that green is not showing up great there, but I've gone over the DNA, RNA, protein steps multiple times, so hopefully it's kind of sticking now. So details of the nuclear envelope, we aren't going to go into the specific structure of the nuclear core. Again, kind of outside the scope of this class, I feel. Okay, so the nucleus. So I keep talking about how chromatin in DNA is very similar. And so here you can see the nucleus contains hereditary of the cell called genes. So here you can see the DNA double helix, and it's getting more tightly packaged around things called histones. Again, I don't necessarily think you need to know to this level of detail. Those nucleosomes get packaged into what we called chromatins, so that's how it was labeled on the previous slide. And that chromatin fiber is even more tightly packaged to what we call a chromosome. So these are kind of different levels of organizational packaging. It's almost like if you had like just a necklace, and then you were going to start to more tightly wrap it again and again and again, and eventually that necklace kind of became a different structure, maybe almost like a rope, instead of just like a tiny little string of beads necklace. You could almost use it as a rope. And again, that's kind of a very similar increase in structure, increase of density that you see with DNA as well. So when you think of when you think of chromosomes, you probably think of kind of these structures that you see here, these X structures. They're connected at the center, which is called the centromere. So when we get to talking about cell replication, that's important. The centromere is because that's where everything kind of gets anchored. And then each kind of leg of that X is called a chromatid. But again, this is kind of, I feel like, outside of the scope. You don't need to get down to this level of detail because this is not a cell biology class, but just recognize that these are kind of semi-interchangeable terminologies for DNA. So chromatid, chromosomes, all comprised of DNA located in the nucleus. If we go back and look at this original set of nucleotides that make up the DNA, there are specific regions within that, kind of stop and start areas, and those are that make code for specific proteins, and those are called genes. So let's say gene 1 would code for, let's say, protein 1, and gene 2, further down that stretch of DNA, would code for protein 2. So that's kind of how the genes are separated, as by what proteins they end up coding for. And again, we've got this beautiful image showing the different chromatids, chromosomes, and the centromeres on those chromosomes. So that's what chromosomes look like. What's interesting is chromosomes only look this condensed and this like very nicely kind of organized when the cell is dividing. Most of the rest of the time they're very loose, and they're being read and they're being expressed and things like that. But when cell division happens, they get really nice, tightly packaged like you see here, because the cell's getting ready to pull those two chromatids apart into two different cells. So you don't usually see chromosomes that look like that, but again, another conversation for another time. Okay, protein synthesis. So I feel like we've talked about this quite a bit as we've been going over the parts of the cell. So, but here we're going to talk a little bit more about the nomenclature. So inside the nucleus, we've got the blueprint for proteins in the DNA. So you've got DNA, your double stranded structure here. What happens is that DNA gets made into RNA. If you want to get specific, it's actually called messenger RNA. And that process is actually called transcription. So you're transcribing DNA into RNA. Once that transcription process happens in the nucleus, that RNA exits the nucleus and goes out into the cell, and then it is translated. So the cell is translating that RNA message into protein. And so here you can see that RNA is being translated into protein via the ribosome. We've talked about how the ribosome is a little protein making factory. And so these are the steps in the process. So in biology, they call this the central dogma. It's very important. If you are going to be a biology major and take any cell biology classes, you'll probably go over this extensively. So again, DNA to RNA is called transcription. I'm going to run out of room here. Transcription, and then RNA to protein. And this is definitely something you should maybe draw out yourself just to keep it intact. That's called translation. Translation. Think of translating nucleic acids into protein. That's kind of where that translation comes from. Transcription and translation, those two words look kind of similar. Just spend a little time trying to keep them separate from one another and not interchange them. The DNA to RNA process all happens within the nucleus, and then the translation of RNA into protein happens in the cytoplasm where those ribosomes are. So again, not the most beautifully drawn central dogma, but you can probably do better on your piece of paper, but it is worth drawing out because it's important. So this is how proteins are made within the cells. Okay, so transcription occurs within the nucleus and is the process in which genetic information encoded by DNA is copied into a strand of RNA to direct protein synthesis. This figure I feel like is a little bit more complicated than we need to be at this point, but it does show you this strand of DNA, the transcribed RNA. This is basically just showing you that the DNA has to open up, that double strands have to open up so the cell has access to the inside of them and it can be read. And then this strand of new RNA is made. So this is your RNA right here showing you the newly synthesized strand of RNA that's going to get sent outside the cell. The RNA, as we've talked about in the past, is composed of slightly different nucleotides than DNA. RNA has uracil instead of thymine, and so that would be one way that you could tell a stretch of RNA from DNA. It also exists in a single-stranded structure, as you can see here, versus the DNA, which is double-stranded, as which you can see here. This process that you can see here is just basically a cutting out of unnecessary RNA that's called these two different regions are being spliced together. This is called splicing. But again, I don't think we need to get into that level of detail for this course. Just recognize that DNA is the template for RNA, and that the process of which this occurs that gets changed from DNA to RNA is transcription. I am not going to ask you to recreate or to redraw or to label a figure at this level of detail. I think that that's maybe, again, a little outside of the scope. Just know what transcription is. And then next we have the process of translation, and this is where you have RNA that's being made into protein. So as we said before, you've got your large subunit and your small subunit and your mRNA that comes in the side and is read, and as it's being read, you have this newly formed protein that's coming out the side here. I don't think we need to get into the level of detail of knowing these sites. So just recognize again that you've got RNA that's being made into protein, and that process is called translation. Just keep translation and transcription essentially straight in your mind, and recognize that it's ribosomes that mediate this process. That's the level of understanding that I think we should be keeping it at this point in time. The EP&A site, if you want to talk about that, if you're taking another class and you're confused by the process of translation, always happy to talk more about translation for sure. Oops. Okay, so transcription, translation. We are not going to go over this slide. I'm going to omit this slide. I'm going to go back and take this out of the PowerPoint lecture. This is, again, just to a level of protein synthesis that you do not need to know for anatomy and physiology. Okay, so really quickly, so we talked about transcription, translation, and so if we're talking about the process, about the central dogma, we've got DNA makes RNA makes protein. Right? We keep talking about this protein. So this process here is transcription. I think I'd get better. This process is translation. I'm just going to write trans here. And then if you have DNA, making more DNA, that's the process of replication. replication. And so this is kind of the first step, I guess, of the central dogma, where you've got DNA being made for more DNA. So you've got this double-stranded DNA that opens up, and you can see here there's a newly synthesized strand of DNA. There's another newly synthesized strand of DNA. So basically DNA is able to copy itself by using itself as a template. That's how you make sure that you have a nice copy of it, as you're just using the original to copy from. Just recognize that you have a double-stranded structure here, and then down here you also have a double-stranded structure. Because you're using your old strand of DNA as a template, this is called semi-conservative. So if you're talking to a geneticist, you'd be like, yeah, DNA replication is semi-conservative. We can talk more about how it's bidirectional, how it occurs in two separate directions, but you'll know that this is two strands of DNA given its composition. Again, it's going to have thymine instead of uracil. So if you had uracil there, you would say that it's RNA, but you don't, so it's DNA. And so this is the way in which cells make more DNA prior to the cell dividing and making two copies of it. So before a whole cell is able to divide and bud and make, or not bud, able to divide and go through the process of mitosis and make two separate daughter cells, it has to make copies of all of the DNA that it has. So each daughter cell has that information that it's carrying with it. So replication is important for that. Okay, so if you're going through your own PowerPoint here, this is where you could click onto the protein synthesis. Again, the process of ribosomes making proteins and extending that amino acid chain, I'm not going to get into. I'm not going to test you over, just because I feel like it's a little bit more in depth than maybe we need to know for this particular course. Cell division. So cell division is a process by which cells reproduce themselves. So we talked about this just a few slides ago. If we look at the cell cycle, maybe some of you who've taken cell biology before have seen the cell cycle. So you have the cell generally just hangs out in interphase for the most of its life. You can see here that in G2 phase, the cell is going to continue to grow. You're going to have other basic copies of proteins that synthesize. Replication is completed during this process. So before the cell even divides, it makes a copy of its DNA. And then kind of the interesting phase that we'll be talking about now is the mitotic phase, where you have this one cell that becomes two cells, two different daughter cells. And so after that, once you have these two daughter cells, they continue the cycle of just becoming metabolically active, duplicating organelles and cellular components, and replication begins. S phase is where your DNA is replicated. Again, this is part of interphase where the cell mostly hangs out. Sometimes cells will exit to the G0 phase, and then if that's a non-dividing cell, like some nerve cells are, you'll just see that them stay in G0 for the rest of their lives. But we're going to kind of just focus on specifically the mitotic phase that you see here. Yeah, and that's for cell replication. Okay, so interphase, like we saw in the last slide, is composed of the G1 phase, the S phase, and the G2 phase. S phase is when DNA replication occurs. It's kind of a big thing in the cell, big occurrence. And we can see that interphase, during that process, you've got the centrosome and the centrioles and the perinuclear material. You see that they're all kind of intact, not really doing much at this point in time, just existing. And then once this process of mitosis starts, they're going to become much more active players, especially those centrioles and of the centrosome. So mitosis is when the nucleus divides. It occurs into two sets of chromosomes and two separate nuclei and has 4 steps. So you have prophase, metaphase, anaphase, telophase. And so we're going to go through each one of these steps and what happens in those steps during the process of mitosis. If you have a hard time memorizing these steps, you know, you can always come up with some type of, I guess, analogy or PMAT. There's also in some textbooks a prometaphase here. So prophase, metaphase, anaphase, telophase. At my last institution, I would always have students come up with memorization techniques here. I think one person had, please pay my absurd tuition. So pay my absurd tuition might be the way that you remember this. There are many many ways in which people can remember the steps to mitosis. So mitosis, the first step would be prophase. So during prophase, chromatin condenses, the chromosomes in the nuclear membrane disappears. So you can see here, you've got your nucleus starting to dissolve. And again, that's important because that nuclear material has to be separated into two separate cells. And so here you have your centromeres that are becoming more apparent. And so during prophase, these highly condensed chromosomes, you see the nuclear envelope dissolves, and then you have those chromosomes becoming associated with microtubules, and those microtubules are going to be what helps pull those chromosomes to each side of the cell as cell division occurs. So those microtubules are anchoring to those chromosomes at the kinetochore, and then once the cells kind of come apart, those legs of the microtubules shorten and pull those chromosomes to each side of the cell. So prophase, again, you've got the nuclear membrane disappearing and the centrosomes moving to two opposite poles to pull that cell apart. Metaphase is probably the most kind of striking phase of mitosis, because during metaphase you have all the chromosomes kind of do roll call and they line up towards the center of the cell. So here you have the metaphasal plate where all the chromosomes align, and because the chromosomes have aligned, they can be, each individual one can be separated and pulled to each side of the cell. So metaphase, like I said, is the most visually striking of all of the phases of mitosis, because it's showing you that there's that roll call to the center of the cell, and then it's going to get pulled apart, and you're going to have equal amounts of DNA into each daughter cell as the two cells divide. So it's important that everything kind of gets lined up before the division of cells occurs, and this is when the DNA is in its most condensed state. Anaphase, the next step, is when those two, the centromeres of the chromosome split, and the sister chromatids, or the two pieces of DNA, go to each side of the cell to the opposite poles. So here, this is where you're seeing those two pieces of DNA that have been replicated are going to be pulled to each side of the cell. And again, very visually kind of apparent. Metaphase, you've got everything lined up in the middle. Anaphase, you got everything kind of moving to each side of the cell, and it's starting to look more and more like two actual separate cells. During telophase, you have what's called a cleavage furrow that appears. This is kind of just showing you down the center of those two cells where there's this little kind of contractile ring that's going to pinch those two cells so they become actually separate. And what's going to happen here is you're going to have the cells kind of looking more normal. You're going to have the chromatin appearance, and then nuclear membrane is going to form again. So as that nuclear membrane was dissolved for this process to happen, now it's going to start reforming so the cells can kind of appear more like they would during interphase and when they're not undergoing mitotic cell division. And lastly here, kind of the step that we didn't note earlier is cytokinesis. Cyto meaning cell, kinesis, being the generation of. After a cleavage furrow forms and the cytoplasm, the parent cells fully splits. When this is complete, the interphase begins. And so again, during anaphase, you've got this cleavage furrow that starts to form here, and once it gets completely cut, you have two identical cells in interphase. You've got a completely formed nuclear membrane, and many of the organelles have replicated, and so the cell can carry out its normal function. It's no longer in the process of cell replication. Okay, so just quickly running through kind of the steps here. Interphase is the period between cell divisions. G1 phase is a metabolically active cell, duplicates most of its organelles and cytosol components. S phase is important because this is when replication of DNA occurs. And so these are kind of subphases of interphase. So replication of DNA does not happen like during mitosis, it happens prior to mitosis. So you can think of the cell as basically prepping itself for this process to happen. So it's going to start making copies of all of its organelles before it actually goes through the process of cell division. In mitosis itself, you've got mitosis, prophase, metaphase, anaphase, and we could even add cytokinesis, which I'm guessing is on the next slide. Mitosis is the distribution of the separate nuclei into two different cells. Prophase, the chromatin fibers condense, nuclear envelope disappears. Metaphase is you've got the aligning of the chromosomes along that metaphasal plate. And anaphase, you've got the centromere splitting and identical sets of chromosomes move to opposite sets of the poles. Again, post-replication, you want to have two identical copies of all chromosomes into those daughter cells. Telophase is when everything kind of starts to return a little bit back to normal. You've got nuclear envelopes and nucleoli reappearing, and chromosomes that reappear, chromosomes and chromatin form, and mitotic spindle disappears. And lastly, cytokinesis is going to be when the cytoplasmic division or the contractile ring forms, the cleavage furrow around the center of the cell, and you've got two individual kind of offspring cells that are made. So you want to have equal portions of those daughter cells going into each cell. Okay, so this is slightly different from mitosis as meiosis. We're not going to go super specific into this. Meiosis is different in the fact that instead of having at the end two copies of your chromatids here, you're going to have single copies, and so the steps look very similar because you do have metaphase, anaphase, and telophase, but your final products are going to have single copies of these chromosomes and not pairs of them. Okay, and again, if we compare these processes here, you can see the first part of meiosis looks very similar to mitosis. And then meiosis 2, instead of having those dual pairs, the end product here only has a single set of pairs. The creation of these are what are called gametes. These are sex cells. Your reproductive cells are sex cells. And so these gametes only have one copy because you want to fuse that sex cell with another one copy of a sex cell, which gives you two copies, and that's what makes a fertilized zygote. So again, having that one copy is important. You don't want to have two copies or you're going to have some issues with reproduction, but we're not going to go into the specifics of meiosis right now. So what's important to recognize is that it's interesting that every single cell in your body has exactly the same DNA, but the way in which that DNA is read and the types of cells it creates are very different. So the DNA in a sperm cell is the same as the DNA in a smooth muscle cell, is the same as a nerve cell, is the same as the epithelial cell, but the way in which that DNA is read or expressed is very different, which gives you all of these different structures. And so when we're talking about the different cytoskeletal components, you can see how those different cytoskeletal components can give you very different cell structures. As we age, our cells deteriorate in their ability to function normally. They're not as responsive, and the numbers of your body's cells decrease. For example, all of the neurons in your brain are slowly going to decrease over time. You get to kind of a peak amount number, and then over time they decrease. There are a few places in your brain where your cells regenerate, but for the most part, kind of what you make as an early to late adolescent is kind of where you peak out and then you slowly degenerate across time. So we lose the integrity of our extracellular components of our tissues, and so again, the wear and tear of our cells. Every time we go through the process of cell duplication, the DNA in our cells get shorter and shorter and shorter, and that's kind of the cellular mechanisms behind why our cells age. Things that can occur when cell division goes wrong, let's say mitosis just kind of goes unregulated and you see cells replicating themselves on an unregulated manner that actually leads to things like cancers. So things like melanoma, sarcoma, osteogenic sarcoma, these are all different cancers of different cell types, of different cell tissues. And so again, unregulated mitosis is what leads to these cell growths, and they're no longer functional in the tissue types that they should be. And we'll talk more about cancers when we get to each one of these tissue types. Growths and spread. Again, some of these cells can actually go from the area in which the cancer was originated and metastasize and go to a different region, so they can grow and spread into different regions of the body and cause those growths in different regions. In terms of causes and treatments, causes can be from genetic or environmental. Again, any type of genetic mutation can introduce kind of this dysregulated cell growth. In terms of treatments, obviously chemotherapy would just disrupt the cell cycle, stop these cells from replicating, or radiation would also just kill these cells. In a more selective manner. The thing about chemotherapy is it's stopping cell division for all the cells in your body. So that's why there's so many side effects seen with cancer treatment. So right now, the way medicine is going, it's trying to become far more targeted, far more specific. So if we're able to identify which cells are cancerous, we can hopefully target those cells for cell death. And so that's kind of where medicine is going right now, is we're going to hopefully target the cells that are undergoing replication in a dysregulated manner and just selectively stop that from happening. Okay, so this week, make sure you're answering study guide questions for chapter 3. Make sure you're completing quiz 3 by Friday. Make sure adaptive practice for chapter 4 is done by next Monday at midnight. And that's all I have for you today. Hopefully you enjoyed this lecture. And if you have any questions or concerns, please do make sure that you reach out. And I'm happy to answer any questions you might have. Hopefully I didn't go too quickly, but that's the advantage, I guess, of having this recorded, is that you can come back and rewatch and pause and re-listen to maybe some areas that seemed a little bit more confusing. But if you have any questions about content or course execution, please reach out. Thank you so much.